the PhD program. So this is human genetics PhD program in the area of home area of genetics and genomics. And we were super happy when she joined the program as a PhD student. And then I've been following through Nicole's work as a thesis committee member, and I've seen the growth that she's taken during these years, and it's been just wonderful to see how she's been developing herself to this independent investigator who is able to think critically, present really well, write outstanding, unbelievably good text, and see her really become this investigator that we want them to become at the end of the program. So that's been fantastic. Nicole also received this very prestigious NIH F31 grant, and that has been funding her for the last couple of years. And that's also a sign how important her work in this field is. And so she's also been very productive and published several papers, and today we will hear about those papers. Welcome to Starting Point.
Thank you so much, Ivy. Thank you all so much for coming. There's so many people here. I'm so excited. We've got a few people on Zoom as well. Oh, more than a few. Okay. A lot of people on Zoom too. Thank you all for tuning in as well. So we're going to go ahead and get started. So you're joining me today for the defense of my dissertation. So my actual written dissertation, which Ivy mentioned, is over 100 pages long. And it represents only a fraction of the work that I completed over the course of my PhD. But I only have 40 minutes. So today I will present a brief version of the 5 chapters. My presentation today, in the simplest terms, is about genetic predisposition to prostate cancer. And I want to start with some background on the field of genetics and how it is thought of in the context of medicine. So let's jump right in. So at the center of genetics is, of course, DNA. Yeah. Ooh. Alright, give me 1 second. Ooh, there you go. There we go. Okay. So at the center of genetics is DNA. 60 years ago, we figured out that the DNA sequence is a set of instructions on how to make other molecules, much more complex ones that can take on thousands of different shapes and functions. They're called proteins, and cells use them to build their own cells, to transmit and receive information, to organize themselves into tissues and organs, and ultimately to create the layers of biology that become an organism. Incredibly, these instructions are flexible enough that they can vary. The sequence is slightly different in each individual, making every person biologically unique. And this information is passed down from parents to offspring, with the DNA from each parent combining into a new sequence in the child. The implications of this pathway are immense. If all that makes an organism can be traced back and distilled into one sequence, then technically, in order to predict all of human biology and to predict any biological ailment in an individual human, all we really need to know is their DNA sequence. This would make DNA the most comprehensive biological marker or biomarker of human health. So this was and still kind of is the beautiful but naive dream of genomic medicine. We started sequencing human genomes in earnest almost 3 decades ago, and we still haven't been able to make inherited genomics a ubiquitous part of medicine. And that is because for 3 decades, we've been learning that things just aren't so simple. One layer of complexity is that the organism is constantly interacting with external inputs coming from our environment, what we smoke, drink, and eat, how we socialize, exercise, and sleep. All these things affect our biology as well. Most health traits are determined from a combination of these factors. The good news is that your environment is often something you can change. This means that you aren't locked into a single fate by your genetics. The bad news is that the accuracy of any predictions we make from genetic information alone is fundamentally limited by just how much the trait depends on genetics versus something else. This means that the genetic biomarker is more effective at predicting traits that have a proportionally high genetic component. The technical term for this genetic component is called heritability. Now, I've told you this so that you have the context to answer the question that I get asked all the time: why do I study prostate cancer? And the true answer, it's because the lab I joined happens to study prostate cancer. But the much more interesting and equally true answer is that prostate cancer happens to be a common disease that also happens to have a really strong genetic component. So how do we know that prostate cancer is heritable? Well, by studying twins, we can actually get an upper bound estimate for how much of prostate cancer risk could be attributable to genetics. So this graph shows these heritability estimates among various cancers, and you can see prostate cancer close to the top with 57%. A small part of this percentage can be attributed to just a handful of genes which have very strong effects. But these variants are very rare. Prostate cancer is very common. So in the vast majority of people who don't have a rare mutation, their genetic risk comes from common variants. We've discovered hundreds of common variants that are associated with prostate cancer risk. Each one has a really small effect, but as an aggregate, they account for a good portion of this heritability. The summed up effects of all these variants in an individual is called a polygenic score. But again, this score is ultimately just a prediction, a probability based on at most 57% of the variables that could affect their true biology. Even a high polygenic score does not guarantee that you will get prostate cancer. Another obstacle to the application of a biomarker is whether the information is actionable. So sure, we can make a prediction about prostate cancer risk, but will this information actually be useful? And I think it will. And to understand why, let's talk about the problems we are trying to solve in prostate cancer. The prostate is an organ in the male urogenital anatomy, located just below the bladder. When prostate cancer is first detected, the vast majority of the time it is localized, meaning confined to the organ, not having spread anywhere else. The good news is that localized prostate cancer is considered curable. Surgery and radiation are both very effective treatments, and men with this diagnosis tend to live for quite a long time. So this bar graph shows the percentage of men who lived 5 years or longer after their diagnosis, and as you can see, it's 100%. In fact, most men live much longer. The 15-year survival estimate for localized cancer is close to 97%. And some men never even require treatment. The outcomes for regional cancer are a bit worse, but the truly dangerous diagnosis is metastatic prostate cancer. During metastasis, cancer cells spread to other places in the body and become much more difficult to treat. Among men who already have metastatic prostate cancer when they are diagnosed, fewer than 40% live longer than 5 years. A small fraction of men start out with localized cancer, which eventually becomes metastatic, and the 5-year survival rate for these is closer to 50%. So here's the dilemma. We need to be able to detect prostate cancer early before it metastasizes, while we can still treat it. But when we do detect localized cancer, we don't actually know which one is it going to be, the indolent kind or the aggressive kind. Now you might be wondering, why not just treat every single case? Well, treatment comes with adverse side effects, primarily by interfering with urinary, bowel, and sexual function. So the consequence of overtreatment of men who do not require it is a worse quality of life. So a key clinical problem in prostate cancer is accurate identification of indolent and potentially lethal cancers at diagnosis. The way we approach this problem is by using the information we have to make predictions about each patient's level of risk. We then try to make management decisions to balance this risk against quality of life. Risk assessments happen at multiple stages of the prostate cancer diagnostic pathway. For example, one way to improve early detection is to perform screening. In prostate cancer, the decision to begin screening and the frequency of the screening test is informed by the risk factors, age, race, and family history. If a man meets specific criteria, he will begin regular testing for the PSA protein, which can be detected from a blood test. PSA stands for prostate-specific antigen. It's naturally secreted by the cells of the prostate into the prostate gland, and when a tumor is present, a lot of the time, more PSA leaks into the blood. But high PSA levels are not diagnostic. This test is prone to false positives, so a biopsy is required for diagnosis. But biopsies are pretty involved procedures, and they come with some risks, so in order to decide whether it's worth performing a biopsy, we can gather some more information with less invasive means and do another risk assessment. If the biopsy is positive, we must decide on the appropriate management or treatment strategy, and that requires yet another risk assessment. Now earlier, I made the claim that the problems we face with prostate cancer are actionable. So every blue box here represents a potential action. Every yellow diamond represents the information that is gathered in order to make a prediction that will inform this action. And throughout my PhD, I've been investigating how adding genomic information could potentially improve the risk predictions made to inform these decisions. 2 of the chapters in my dissertation are dedicated to these contexts. I will talk about risk stratification for screening in the next chapter, and risk assessment before treatment in the 3rd chapter. In the 4th chapter, I will change course a bit and talk about some software that I developed to help with these applications. Now let's get started with the first research project. This is a collaboration with the team at UT San Antonio, led by Dr. Mike Liss. Liss is really interested in risk stratification prior to biopsy to make sure that only men who will actually end up needing treatment have to go through this procedure in the first place. Our most promising tool for this is screening for high levels of PSA. But as I mentioned earlier, PSA is prone to false positives. One way to adjust for this is to tune the frequency of screening based on a risk assessment. Currently, men who decide to begin PSA screening are screened less frequently if their baseline is below 1 nanograms per milliliter, because studies have shown that this baseline level is associated with really low future risk of prostate cancer. But if you're above 1, but below the level required to trigger a biopsy, there's no further stratification. We thought this could be a good context to use a genomic risk estimate to further stratify men according to their future risk of cancer and qualify some men for more or less frequent screening. Specifically, we wanted to use a polygenic score, or PGS. I mentioned this concept when introducing heritability. Each of these arrows represents a common DNA variant that is associated with getting diagnosed with prostate cancer within a lifetime. They each have a small effect, which has been statistically measured in association studies with hundreds of thousands of participants. To get an aggregate measure of polygenic risk for a single person, we multiply each effect size by the number of variant copies this person is carrying. We then add up these weighted effects into a single score. Since everyone has a slightly different combination of variants, every person will have a slightly different score, and we can place all of those scores on a distribution. People with the highest scores have the greatest polygenic risk of prostate cancer and vice versa. Now back to the study. Now we didn't have a lot of money, but we had enough to get the genetic variant information, or genotype, from 310 participants in the San Antonio Biomarkers of Risk study, or SABOR. For this program, when men who don't have prostate cancer come to the clinic, they are asked if they want to begin PSA screening and participate in research. If they consent, their clinical information is collected for as long as they're in the health system and is made available for researchers. Since we could only get a small sample size, we wanted to very carefully choose our analysis groups to give us the best chance of a statistically powered analysis. So first, we looked at all the men with baseline PSA greater than 1. Then we defined 3 groups. First, men who are diagnosed with clinically significant cancer. That's cancer that, according to current guidelines, is considered necessary to treat. Then, men with diagnosed with low-risk cancer that is recommended for surveillance. And finally, men who never got diagnosed with prostate cancer after a long time in the study. These are our no-cancer controls. We wanted to account for known pre-biopsy risk factors like age, race, family history, and PSA. To do this, we used an existing risk calculator to predict the risk of clinically significant cancer based on these variables. Then we matched the groups on these scores. This process of matching makes it so each group has the same distribution of these known risk factors, which also means that any other differences between the groups must be caused by something else, for example, a genomic difference. Next, we computed a PGS in each subject, combined the scores into a distribution, and separated the cohort into 5 quintiles. This graph shows the proportion of each prostate cancer category in each quintile. Now remember that the highest quintile represents the subject with the highest PGSs, and therefore the highest measure of genomic risk for prostate cancer. So we would expect the highest quintile to have a lot more men with prostate cancer than in the lowest quintile. And that is exactly what we see. So the purple and green sections are both men with prostate cancer, and you can clearly see an increase. Proportionally, the no cancer controls start out big and get smaller. If we look at just the purple sections, we also see an enrichment of clinically significant cancer among men with higher scores. As much as I like looking at this graph, observation should be supported by statistics, and we did do some. So this chart shows the results from a multinomial logistic regression. This regression models categorical predictors and categorical outcomes. On the y-axis, we have the predictor, each quintile category. On the x-axis, we have an odds ratio. Each quintile shows the results of 2 comparisons, clinically significant versus no cancer, and low risk also versus no cancer. The top result here represents the following case. If we only consider 2 possibilities, clinically significant cancer in purple or no cancer in yellow, you would have a 7 times greater odds of being diagnosed with clinically significant cancer if you had a score in the 5th quintile versus the 1st quintile. It's a mouthful, but this statistically confirms that the purple to yellow proportion is bigger in the 5th versus the 1st quintile. We also see a significant but slightly smaller difference in the green to yellow proportion. So let's put this in context. We want to see if we can isolate a group of men that could benefit from more or less frequent screening. If we use a low percentile threshold of the score, this creates a pool of men where the majority will not get cancer, making them great candidates for low frequency screening. Vice versa, if we use a high percentile threshold, we get a pool of men enriched for those who are at risk of clinically significant cancer, which makes them good candidates for high frequency screening. But we face a problem here, which we see with a lot of biomarkers. Every quintile has representation from every category. That means there will always be a trade-off where some fraction of men may get underscreened or overscreened. We ran a couple more analyses to further support these findings. If you're curious, you can read up on all the details in our paper. And we came to the following conclusions. First, it is clear that the PGS contains information about risk of prostate cancer that is not already encompassed by other known risk factors. This means that it would be useful to incorporate PGSs into risk calculators like the ones we used to match our cohort at the beginning of the study. Second, it is also clear that it is possible to use a percentile threshold of a PGS to create a pool of people who are enriched for clinically significant prostate cancer patients, and vice versa. That's actually fairly impressive given we started with a pretty small group of people. Third, and this point was better supported by other analyses, the PGS is not very effective at separating clinically significant from low-risk cancer. So it is not a perfect biomarker for this particular task. But we do have a good proof of concept here that a PGS could be used to target screening frequencies. The next logical step would be to properly assess the impact of the trade-offs that would be introduced. We would need to do this in a larger cohort that is more generalizable to a typical screening population, since our matched design was not representative of a real-world risk distribution. It would cost more money to do this, but smaller, cheaper studies like these show that there is reason to continue investigating. And with that, let's move on to my next project. And I will take a sip of water. So for this project, I was able to study a phenomenon called upgrading. This topic is relevant to the risk assessment that occurs after you take a biopsy of the prostate. A biopsy provides an important piece of information, the grade of the tumor. A grade is often given by a pathologist after they look at a section of the tumor under a microscope and see how much the cells have changed from what they would normally look like if they were healthy. They are graded on a scale, and higher grades correlate with greater risk of metastasis. Higher grade tumors are almost always treated aggressively, but low grade tumors are considered low risk and usually qualify for surveillance where they are monitored. But there's a catch. Every once in a while, a low-grade patient will choose to get a surgery soon after their first biopsy, which gives the pathologist another chance at grading the tumor, this time by looking at the entire prostate. And in about 20 to 30% of these cases, the second grade actually ends up being higher than the original grade at biopsy. This phenomenon is called upgrading. This could be happening for two main reasons. The first is that prostate cancer tumors are notoriously heterogeneous. It is possible that the needles from the biopsy did not catch the part of the tumor that was of a higher grade. The other possibility is that during the short time between the first biopsy and the surgery, the tumor had actually evolved to a higher grade. Both of these outcomes mean that grade-based risk assessments are sometimes inaccurate, and some patients may be needing treatment sooner than when they're actually getting it. If there was a good biomarker for upgrading, we might be able to adjust these risk assessments accordingly. So I had the opportunity to look for such a biomarker in collaboration with, once again, Dr. Mike Liss at UT San Antonio, Dr. Robin Leach, who coordinated this study, and the Early Detection Research Network, or the EDRN. The EDRN is an interesting organization. They're at the National Cancer Institute, and their job is to facilitate biomarker discovery. One of the things they do is they curate blinded validation reference data sets where a biomarker can be evaluated. Robin coordinated an effort to create an upgrading reference set at the EDRN. These are 285 men diagnosed with grade group 1 prostate cancer who decided to undergo a prostatectomy within 2 years of their diagnosis. The controls of this data set are men who did not experience upgrading after surgery, and the cases are the ones who did. Of this cohort, we were able to sequence the genomes of a subset of 192 men, so not the entire cohort. And another important note is that this data set is blinded, which means that researchers like me can work with this data, but we do not get to know which samples are controls and which samples are cases. The clinical team chose a variety of potential clinical biomarkers for testing and had the EDRN team fit a multivariable regression model to see if upgrading could be predicted. Two variables were statistically significant: PSA density, which is PSA divided by the volume of the prostate, and the variable indicating the average percentage of cancer tissue in positive biopsy cores, or cancer percent as it's abbreviated here. We then took just these two variables, fit another regression model, and computed a prediction accuracy measurement called the receiver operator curve. This curve compares the false positive rate and true positive rate of predictions made by the model at every possible cutoff point for a binary prediction. The area under the curve represents a unified measure of accuracy for the model, with 50% indicating a 50-50 chance of getting a prediction right. Here, we got 75% for correctly predicting upgrading status, which is pretty good. Now, it was my job to come up with a genetic biomarker to test in this cohort. First, I looked for known rare pathogenic mutations in genes that are involved with the DNA repair processes. These have been repeatedly implicated in cancer risk. The gene with the most evidence for prostate cancer is BRCA2. And here I ran into the common obstacle when studying these mutations. They're very rare. I identified only one person in the cohort with the BRCA2 mutation. You can't really do a statistical test with one event. So I switched to the polygenic risk score. I computed a PGS for prostate cancer in every subject and sent it to the EDRN to test against the cases and controls. And here's what I got back. Now this is what we call a negative result. This curve is essentially just a diagonal line, which means that the prediction made by the PGS was equivalent to flipping a coin, accurate only 50% of the time. Now, it could still be that there is a small genetic effect, and we simply don't have a large enough sample size to detect it. But even if that were the case, it would not be very practical to use, given that we've already discovered some much more effective clinical predictors. But there was one more genetic trick that we wanted to test with this cohort. We had shown before that PSA density was strongly associated with risk of upgrading. The interesting thing about PSA is that some folks have naturally higher baseline levels of PSA. These people are actually at higher risk of a false positive screening test because their high PSA levels are not indicative of a tumor. They're just naturally high. Even more interesting, some of this natural variation comes from a genetic predisposition. We hypothesized that this natural variation may also be reducing the strength of the association of PSA density with risk of upgrading, and we could improve this association by adjusting for its genetic component. So another group had actually discovered a handful of genomic variants that are associated with this baseline PSA variation in healthy men, and they also published the effect sizes for a polygenic score. So for each subject, we took the original PSA density, this is the formula, and then we divided the PSA value by the value of the polygenic score for PSA, which effectively normalizes the PSA measurement by the genetic predisposition. We sent that off to the EDRN, and they came back with these curves. So one curve is for adjusted, and one is for the unadjusted PSA density. And if there had been a difference in the prediction accuracy, one curve would be wider than the other. These two are basically overlapping, meaning there was no difference. So interestingly, that is another strike for polygenic risk in this study. So let's sum up the conclusions of this project. First, we clearly identified two clinical biomarkers of upgrading, PSA density and cancer percent in positive biopsy course. Second, I showed that the genetic component that affects baseline PSA does not interfere very much with this prediction. I also showed that a PGS for predicting the diagnosis of prostate cancer was not very good at predicting upgrading. This implies that a higher polygenic risk of prostate cancer in general does not preferentially predispose to upgrading. If we think about this from a biological perspective, it implies that most of the variants in the PGS are not directly involved in biological pathways that lead to upgrading, such as increased tumor heterogeneity or rapid grade progression. That's mostly just my speculation. Ultimately, the greatest benefit of the study was the creation of a validation data set at the EDRN, including processed genetic data, courtesy of yours truly, which gives other researchers the opportunity to continue testing other biomarkers. And now we can move on to my final project. You may have noticed that I have done a lot of work with polygenic scores. And the thing is, I only really get to touch these scores at the end of their journey. So here's what I do. I showed before that a PGS is just a set of variants and their effect sizes. Calculating these effect size values is a really complex technical process. Statistical geneticists have been tackling really interesting problems in order to improve the accuracy of these values. But after the effects are calculated, they're routinely published and freely available for anyone to access, including researchers like myself who are primarily interested in all the different contexts these scores could be applied afterwards. So my task is relatively simple. Let's imagine this is my matrix of samples and genotypes. All I have to do is match up the variants to their effect sizes that are published by somebody else, count the alleles in each genotype, and sum up the columns. It's a relatively simple procedural task. So you would think that there would be a free software tool out there that a researcher with minimal programming experience could use to quickly apply an existing PGS model to their genetic data set. Well, surprisingly, you would be mostly wrong. Yes, there is software that can do this, and here are some examples. These two tools are generalist genetics toolkits designed primarily for that first step of PGS effect size estimation. You need to dig through a mountain of documentation before you find what you actually need, and you kind of need to already be an expert in genetic data handling because the very first step is to convert file formats via the computer command line. This tool is specialized for the application step, but also requires command line expertise just to run the software. So when I realized that I don't actually need these tools for my workflow, I started writing my own code for PGS application, and eventually I thought maybe somebody else out there might find this useful. So I decided to turn my scripts into a formal piece of software. And then I rewrote everything at least twice over. But the result is Apply Polygenic Score, which is a software package for the application of an existing polygenic score to new genetic data. And it is designed for non-experts. It's written for the R statistical programming environment, which is widely used by researchers across many disciplines. It expects the standard genetic data file format. It is extensively documented, and it includes a tutorial. It has three main categories of functions: those for pre-processing your genetic data, those for the main PGS application process, and a set of plotting functions that automatically generate visualizations based on the outputs of the previous functions, and these show the quality of the scoring and the result of automated analyses like correlations and those receiver operator curves. When you publish software, it is generally expected that you show how your tool compares to other similar ones, and this process is called benchmarking. So I benchmark my tool against 3 others, and first I want to show you the accuracy of the PGS scoring. Here we see the same score in the exact same cohort computed by my tool versus the tool Plink, which is more or less the gold standard for working with genotype data. You can see that the correlation is pretty much 100%, so great, we can check that off. But if you zoom in, you'll see that there are a couple of points that are just off of the diagonal axes. These actually represent individuals who have a third allele at a multi-allelic site. This is kind of an edge case in genetic data that messes with the convenient binary encoding of alleles. Most software basically assume that there can only be 2, and Plink is one of those, and it does not correctly handle these types of variants. In fairness, they do ask you to exclude them before you do your analysis, and these sites are not super common, but my tool handles them correctly. For more technical details on the why and the how, I encourage you to read my paper. The second type of benchmarking is resource benchmarking, where you give the software bigger and bigger inputs and compare how much computer memory and time the software needs to complete an operation. So here we have runtime on the left and memory on the right. The input size on the x-axis is defined as the size of the sample by variant matrix. The y-axes are in orders of magnitude, but I've added a label and a horizontal line at reasonable practical thresholds of 15 minutes and 10 gigabytes. The data points from my tool are in purple. And what we see is that for the small to middle range of input sizes, my tool performs about the same as the rest. But as we cross into the really big inputs, my tool begins to lag behind, right about here. So these results tell us the practical limitations of the tool. I wanted it to be really easy to use, so I wrote it for the R environment. R is considered very intuitive. It's designed for easy data manipulation and analysis. The trade-off is that it is slower than many other languages and has very severe limitations on memory manipulation. So I actually wasn't surprised to see these results. But the great news is that this tool is actually perfectly fast and efficient enough for its intended purpose, which is to take existing PGSs, which usually contain on the order of hundreds to thousands of variants, and to apply them in clinical cohorts, which usually have on the order of hundreds to maybe thousands of participants. And for studies of that size, this tool is still practical and convenient. So I think what I want folks to take home about this project is that it's trying to fill an accessibility gap for polygenic research, because it is truly a fantastic achievement that there are PGSs for thousands of traits that have been trained and published, with presumably the goal of sharing this hard-earned knowledge with a wider range of researchers, many of whom simply don't have the resources or expertise to estimate these values from scratch. But as experts in their own fields, they can take them to the next step by testing where our PGS is actually useful and where they are not. And now they can do this without also having to get a degree in bioinformatics. And with that, it is time to wrap up. So genetics is a very broad field. My dissertation is in the branch of translational genomics. I stand on the shoulders of giants, basic science researchers who brought our understanding of genetics far enough for me to be able to take what they have discovered and see how this knowledge can be applied to medicine. All my work has been published in clinical journals. I get to collaborate with doctors who are so excited about the possibilities of genetics to help their patients. It's a very exciting time in our field, and I'm very excited that I got to share a piece of it with you. And I want to say that when I introduce myself as a cancer researcher, people will often tell me about their experiences with cancer, because pretty much everyone has had an experience with cancer, and they want to know if we're making any progress. And I think my favorite thing about what I do is that I'm actually able to answer these questions. I consider it a privilege to work on something that touches so many people, and I consider it a responsibility to be able to communicate what I have learned. It's a skill that I've tried to improve over my time here, and this presentation is kind of a test of that too. So after I'm done being interrogated by my committee, please let me know what you think worked and what maybe didn't for your background and level of expertise. And that is the end for the science that I have to present today, and I have very many people to thank. So it is traditional to start by thanking your PhD advisor, and for about 4 years my advisor was Dr. Paul Boutros. He's not here today for complicated reasons that I do not fully understand, but he taught me to not just to be a curious scientist, but to be a very rigorous scientist. And for that, I am extremely grateful. One of the earliest pieces of advice that I remember receiving from him is that I should never undersell myself. So in the spirit of those words, since he is not here to say it, I think I'll go ahead and say that I think I did a pretty good job with this PhD thing. I want to thank, of course, Dr. Paidi Payukata. She's been my official advisor for the last 3 to 4 months. But as the director of our PhD program and as a member of my committee, you've been a mentor for much longer than that. And I'm so grateful for your patience and support throughout this time. So thank you. I want to introduce and thank the other members of my graduate committee, Dr. Paul Spellman, Dr. Noah Zeitlin, Dr. Amar Kishan. You've always given me excellent advice when I needed advice, and you've always supported me when I needed support, so thank you so much. So many other faculty have been mentors to me throughout my PhD. These are my teachers, my counselors, people who vouched for me, who wrote letters for me, who checked in on me, and I appreciate you all, and thank you so much for being here. Next, I want to thank my collaborators. These are doctors and investigators outside of UCLA who entrusted me with their data, who believed in our projects, and who were willing to both teach me and learn from me. Thank you so much. And these wonderful people are my co-authors from the Boutros Lab. Without their collaboration, I literally would not have a dissertation, and most importantly, I would not have learned nearly as much. Thank you all. So I really love doing research, and I still really love doing research. I want to keep doing research, which is a relief because when I started my PhD, I was pretty scared that I would burn out. It's a tragically common occurrence in our field, and I'm pretty sure that the reason it didn't happen to me is because of the community of amazing people who have supported me this whole time, starting with, of course, my amazing lab mates. These are the most brilliant, capable people you will ever meet. They're always willing to help with any problem, to donate their time if you need it, and crucially, they celebrate each other's success instead of coveting it. Thank you so much for motivating me, for teaching me, for keeping me sane, and for all of the happy times outside of lab too. Thank you so much, Boutros Lab. I want to give a special shout out to my cohort mates from the genetics program, Jennifer and Izzy. We started our PhDs together during quarantine, and somehow we still managed to become and remain close friends to this day. I appreciate you so much. So we actually started rock climbing together back in 2021. And since then, I have been going to the climbing gym at least twice a week, every week, no exceptions, except for when I'm on vacation. I did some back of the napkin math, and I spent over 1,000 non-consecutive hours at the gym during my PhD. I've met so many wonderful friends there, and many of them are watching on Zoom, and some of them are actually here in person. And I'm very sorry if I didn't find a picture of you because I realized that I haven't been taking a lot of photos of the gym. But thank you so much for creating this community where I could take refuge away from all the stresses of PhD life. Thank you to all my friends from college, high school, and even elementary school who have stayed in contact all this time and reminded me that there is a life outside of academia. It has been so exciting watching you become productive members of society while I stayed in school and acquired a completely impractical skill. I am so sorry, guys. I cannot give you medical advice. But if you ever need a genetics lecture on short notice, then call me anytime. Next up to my family. This is what it looks like when we're on our weekly Zoom calls. And this is what it looks like when we're together. So thank you for supporting my decisions, for always making me feel loved, and for tolerating my working through our family vacations. They're all here today. If you see them, thank them for doing great work. I also have a lot of family abroad, some of whom are tuning in. They may have not made it this far because it is the middle of the night for them. But if they're still awake, I thank them very much for watching. And finally, I've been to a lot of defenses, and I can tell you that a lot of cats have been thanked over the years. I do not have a cat. I have a crested gecko. This is Flo when she was just barely hatched, just 6 months before I started my PhD. This is what she looks like now, all grown up, and I think she's adorable. I will not be thanking her for getting me through this because she mostly just tolerates my presence, but I think she's very cute. And that is it, and we can ask some questions now. Question.
How did you decide on R as the language? Were there other options you were looking at?
It is the language that we use ubiquitously in our research lab, and I know a lot of other researchers in sort of statistically and analytical fields who use this language a lot. It's well designed for a lot of data wrangling and statistics type of analyses. So the cool part about my tool is that you don't actually have to go in and out of different environments to do an entire project in genetics necessarily. So you can calculate the plug-in score in R and then port that right into your next regression analysis and then make your beautiful plots and things like that. Any other questions?
I have a question. I'm curious, so you showed some variables that are important for informing treatments or providing medical advice. So how do findings from researchers such as you get incorporated into treatment plans in the hospital? So how does it get integrated in practice?
Yeah, that's a really good question. There are a lot of different ways and a lot of kind of stages along which that happens. The sort of you've made it is when your tool or variable gets incorporated into guidelines that doctors refer to when figuring out how to manage their patients. And those guidelines are updated very frequently based on scientific literature. A lot of the time they rely on, again, the gold standard of medical intervention research, which are clinical trials. So with a predictive or with a screening paradigm or a predictive tool, the gold standard would be some sort of clinical trial that shows that there is a net benefit to using your tool balanced against the cost of the tool and measuring that tool, the morbidities associated with it, quality of life aspects, things like that. It can be a pretty long process. But in the meantime, doctors do have the leeway to read research and make decisions based on that, even if they're not like actually in guidelines, of course after conversations with their patients. So there are ways to make these kinds of things more accessible. For example, like creating nomograms, so like easy or like web calculators, which I know that doctors will sometimes just like pull up on their computers and like put in some variables and see what the predictions are from just based on like the web application, and just talk it through with their patients and what that might mean. Yeah. Dr. Alcott.
Yes, it's great to be here. Thank you. Great talk too. I enjoyed it very much. In the analyses you performed, you included the polygenic scores of prostate cancer in particular, and we can imagine that indeed for the upgrade of the cancer, that the risk of getting prostate cancer is different from the disease progression or the upgrade itself. Have you considered using other polygenic scores, more explorative, to see if there are some other features you could uncover or genetic profiles that may be associated with the upgrade risk?
Yeah, so this would be ideal. Like the ideal way to go about this is to train a score on the actual traits that you are trying to predict, right? But the biggest difficulty with this is that you need a really large sample size and really well annotated cases and controls, or maybe in this case, case versus case analyses. So at the moment, there simply like don't exist prostate cancer case pre-trained scores that have been done in large enough cohorts for me to use in these cases. And I could attempt to do that on my own, but my cohorts have been generally so small that it would be unlikely that I would be able to find a signal on the genome-wide scale for something like this.
I didn't mean for discovery, I mean for application. Let's say breast cancer or other types of cancers may harbor also the risk for disease progression in your case, so there's a difference. Those scores are available from other large-scale studies.
From like other traits, other cancers, that's pretty interesting. I haven't considered looking beyond the prostate cancer like paradigm ecosystem, but that could be very interesting. Yeah. Yeah.
How common is it for prostate cancer patients to get like their genome sequenced for work like this? Like, is it only people that opt in for research studies, or just can anybody do it?
So, sequencing like from blood samples in inherited genetics is not like standard of care at the moment. It's not very expensive anymore, and we're moving towards it, but there isn't like a standard guideline for which the type of information would be used, and that's part of the problem. And we're just kind of not quite there yet, but we're getting very close. There are other types of genetic information that can be part of standard of care. For example, looking for those rare pathogenic mutations for certain risk indications, that is a standard thing to do a gene panel or look at a very specific gene to see if there's a variant that could have been inherited and passed down through the family. But the kind of large-scale sequencing to get enough information for a polygenic score, for example, is not currently standard. Yes, but the paradigm for sequencing, for example, a sample from the tumor itself, that's a slightly different story.
First of all, fantastic work as a whole, Nicole. I agree that if Paul was here, he would have extensive number of platitudes to say. I think prolific is a good one because you've been, so consistent with the work that you put out. I have a question kind of building on what the gentleman over there on the corner was mentioning. For the upgrade project number 2, I think it was like slide 27 or something like that, where you were saying, explain the data set. I'm a little confused. Could you explain a little bit more if it was whole genome sequencing on just the biopsy itself, or both on the biopsy and the resection?
Yes, let me pull up. Here we go. So the genome sequencing that we did was from blood samples.
Oh, got it. Okay.
Yeah, so this is your inherited DNA, right? So
Got it, got it.
Not from any samples from the prostate itself. I do believe that they saved those biospecimens. I would be surprised if they did not, but it wasn't part of this initial study to go into that type of information.
Ah,
Yeah.
okay. And I guess, so you mentioned a little bit about like power, and kind of building up on the last question is, like, if we were to get to a point where like one day we're able to get a panel and just make it standard, I guess like what would that future look like? Like at what point would you start deploying these like polygenic risk scores or like panel sequencing to kind of be able to tease out if a patient might be more likely on the, fifth level of risk, I guess. You have any thoughts on that? Because you're an expert now. You can comment on this.
So we're getting close. Some hospitals, some systems are already offering this. I think Mass General, I looked this up not too long ago. Mass General offers a polygenic risk panel for cardiovascular traits now already. I think the greatest benefit is the declining cost of sequencing technology, where like it's really like no longer a reach in order to pay for these things, and eventually we need to get to a point where first there is clear medical indication for this information, right? Clear medical utility. Then it becomes part of guidelines, and then it also becomes something that insurance companies are willing to pay for. At that point, that is when something like this would probably become a staple thing. What can it be used for? So many things, right? And the cool thing about your inherited genetics is that it's static, right? It doesn't actually change. So you only need to sequence it once, and then it opens up so many both research, downstream research opportunities to see how that information can be used for other medical applications as well. Did I answer your question? I feel
Yes,
like
you
I
did. No, it's perfect. No, thank you. I think like my takeaway is that I can see how, in the future, as soon as they start doing like PSA, they can start doing like a, polygenic risk score assessment simultaneously, and then just like start,
Maybe an interesting example is what UCLA is doing with their biobank right now, which, if you guys don't know, if you're part of the UCLA health system, you can opt in for any of your bloods. If you ever go into the clinic to do a blood sample for any reason, that sample can be used to sequence your information and be included in the UCLA biobank, which is then available for researchers to use in UCLA studies. So that's if this kind of sequencing becomes like standard of medical care, then that like pot of information becomes bigger and bigger and bigger. And of course, people would need to consent in order for it to be used for research, but there are so many like opportunities for more discoveries to be made with that kind of information.
Thank you. Awesome stuff.
So I guess you guys all have to leave now.